Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh to all my students. Inshallah, today we are going to continue with the third lecture out of lecture series of gastrointestinal tract system. And for today, the title is Lower Gastrointestinal Tract Part 1, meaning that in the past we have already covered the function of upper gastrointestinal tract, upper GIT, which includes the mouth, pharynx, esophagus, and also stomach. So today we're looking into more detail of functions of small and large intestine. If we are looking into this diagram, we can see that lower gastrointestinal tract is referring to the duodenum, which is the first part of small intestine, jejunum, the middle part, and ileum, which is the third part. Then it continued with the large intestine that divided into ascending colon, transverse colon, and descending colon. And there is a sigmoid colon because of the shape like a S shape, so it's known as sigmoid colon, rectum, and anus. So small intestine is referring to this area. So at the end of this lecture, I hope that our students will be able to state functional anatomy of small intestine. How is the anatomy structure actually fit with its function? Secondly, to describe the different types of small intestine movement and motility, basically how the food being moved about in small intestine, and also able to describe intestinal secretion. Now we'll start with small intestine. Small intestine is covered with mucosa. At the top layer of the small intestine is known as mucosa layer, and it is covered by villi. What is villi? It's actually referring to the finger-like projection. So it's quite different as compared to pharynx, esophagus, as well as stomach, in which they have this smooth surface of mucosa. However, in small intestine, there is a presence of villi. This villi is supplied by an arteriole that break out into capillary networks. So blood vessels that actually surrounding the villi, and also a lymphatic vessel, or which also known as lacteal, that occupies at the center of the villus core. This is actually to optimize the ability of this villi to absorb, for the blood capillaries to absorb the nutrients that been absorbed by villi. And this villi also covered by a single layer of columns epithelium, which is actually cells of small intestine. Free edges of these cells are divided into minute microvilli, which that looks like a brush border. So in addition to villi, there is like microvilli, a smaller finger-like projections that actually will further increase the surface area of small intestine. The outer layer of the cell membrane of the mucosal cells also contains many digestive enzymes, and also presence of valvular conniventous, which is valve-like folds in the mucous membranes, and also presence of villi and microvilli increase the ability of small intestine to absorb the final product of food digestion up to 600 times. Okay, so these are the features of small intestine that actually fit its purpose as the main function of small intestine for nutrients or final products of food digestion absorption. And this diagram illustrates to us with more details, like this is the mucosal layer, and we can see that the villi, this is a finger-like projection, and at the top of this villi, this is microvilli, like a brush borders here. Okay, so there is a blood capillaries, that surround the villi, and also the lacteal. Basically, this is the lumen area where the food digestion is taking place, and the end product, like for example, like protein, the end product is amino acid, will be absorbed into the cell of intestine by the microvilli and villi presence, and then at the middle of it there is a blood capillaries and also lacteal for further be absorbed into the blood vessels. So motility of small intestine, if in esophagus and stomach, segmentation and peristaltic are among the mechanisms how the food being moved around. In small intestine, there are two another additional motilities, which are tonic and also migrating. So segmentation is the main mechanism of motility in the stomach. This is actually ring-like contraction, like move and mix the chyme to and fro, and increase its exposure to the mucosal layer or surface, and it helps to chop the chyme 2 to 3 times per minute. Meanwhile, peristaltic is the main mechanism of motility in esophagus, and it moves the chyme or bolus, but in the small intestine, the food present as chyme, move the chyme in the caudal direction along the intestine, and the duration between from duodenum to ileum, the food takes about 2 hours. Okay, and for tonic, it's only present in small intestine. It is actually prolonged contraction, and this permit longer content of the chyme with enterocytes and promote absorption. And migrating is referring to taking place in between meals, okay, and coordinate emptying of the stomach and small intestine. Okay, so and small intestine motility. So looking into this, we can see that in the small intestine, because it's a long tube, it's about 6 meters long, it needs some more, like several types of motility to actually move the food forward down the tract. And in the small intestine, there are also intestine fluid or secretion that been secreted, and it is secreted by two types of glands. The first one is Brunner glands that actually located mainly in the duodenum, that secrete a thick alkaline mucus, mainly to protect the duodenal mucosa from the gastric acid that actually present in the chyme that arrive from stomach. And in small intestine also, there is a presence of intestinal glands, which also known as crypt of Lieberkühn, that secrete an isotonic fluid, like a water fluid containing mucus and few enzymes. So in small intestine, only several enzymes been secreted. Mainly the intestinal secretion composed of water, which is about 98%, and also a little bit solid is about 1.6%, and about 1 liter of intestinal secretion being secreted. It is colorless, and the pH is almost like a normal neutral pH, like 6.5 to 7.5. It is actually to stabilize the chyme that present as acidic. So with this pH nature of intestinal secretion, it can help to reduce the acidicity of the food that arrive from stomach. So the intestinal secretion is regulated, as I mentioned before, by Brunner glands of the duodenum. These actually are compound that tubular submucosal glands found in the portion of the duodenum which is above the hepatopancreatic sphincter. And intestinal secretion is secreted or increased by the presence of fatty food that been passed from stomach, and also presence of hormones, specifically secretin and endocrinin, and activation of vagus nerve that stimulate the secretion of intestinal secretion, and also mechanical stimulation to the mucosa. Basically, the presence of food itself will stimulate the mucosal layer, and then can send message or trigger the for the intestinal secretion to be secreted. And the intestinal glands, which is crypts of lubricants. It is referring to the tubular glands that lie in between the finger-like projection of villus. So between the villus there is a intestinal gland. And the secretion, the aqueous secretion of intestinal glands is increased by the presence of hormones such as VIP, GIP, gastrin, and also glucagon, and distension of the intestinal lumen by accumulated gut, meaning that the presence of the food will cause distension of intestinal lumen, that's giving a signal the presence of food in the small intestine. Therefore, the crypt of lubricant will secrete the aqueous solution. So now we are going into more detail the digestion and absorption process of carbohydrate, fat, protein, and also vitamin and calcium. First, we'll start with the digestion of carbohydrate. In many occasions, we ate carbohydrates as polysaccharides. It means plants polysaccharide, for example, like starch. It also we ingested like rice, bread, as well as noodle. It's partially digested in our mouth with the presence of salivary amylase. Okay, so the enzyme that is responsible to break down the polysaccharides bonds or linkage is salivary amylase. However, because the food only present in the mouth for short time, so it's only partially digested. The digestion process of carbohydrate will be further continued in the small intestine by the presence of pancreatic amylase. So this amylase is not secreted by small intestine. Instead, it is actually secreted by pancreas. Okay, so this polysaccharides chain will be broken down into disaccharides, which actually compose of maltose and also polymers that composed by about 8 glucose molecules, and further will be in small intestine. It will be further broken down into the simplest form of carbohydrate, which is glucose, the presence of maltase enzyme. For sucrose, it will be broken down into fructose with the presence of sucrase enzyme, and for lactose, it will be will be broken down to galactose and with the presence of lactase enzyme. So these are and this is taking place at the small intestine, and this also with the presence of these enzymes. So the end product of carbohydrate glucose is either glucose, fructose, and galactose. Okay. The cellular level, if looking into this diagram, so imagine this is the lumen, which the digestion taking place, in which carbohydrate being converted into the simplest form, either is glucose, galactose, or fructose. So there is a brush border of microvillus, and then here there is glucose is transported into the intestinal cell, that is coupled to sodium transport via the transporter sodium glucose transport, SGLT1 protein. So basically this is the protein channel or protein transport. This is a transport protein that facilitate the transportation of glucose from outside of the cell, from the lumen into the intestinal cell. Okay, so basically with the movement of sodium, it will produce energy that will be used for the glucose to be transported into the cells via SGLT protein. And this sodium is actively transported out of the cells. So you can see here, the sodium will be transported out of the cells, and glucose enters the interstitial by facilitated diffusion via glucose transporter GLUT125. There are several types of glucose transporter depending on the specific type of glucose. So we can see here, it's diffused into the blood capillaries with the presence of energy that produced by the sodium transportation. This is known as secondary active transport, in which glucose is transported into the capillaries. Secondary to active transport, the energy for glucose transport is provided indirectly by the active transport of sodium out of the cells. And then we go into protein digestion. Protein is broken down into peptide fragments. As we know, protein is formed by polypeptide, so there are few linkage or bonds that need to be broken down. Basically, the digestion of protein mainly in stomach by presence of pepsin and also trypsin, and it's further continued in the small intestine by trypsin and chymotrypsin. Protein further digested into free amino acids by carboxypeptidase and also aminopeptidase. So basically, this pepsin is secreted in the stomach in the inactive form, and then hydrochloric acid will convert the pepsin into the pepsinogen. Okay, the pepsinogen into pepsin, which is the active form, and then the trypsin and chymotrypsin is secreted by pancreas. And in the small intestine, it secretes carboxypeptidase and also aminopeptidase. Will further digest protein into the smallest form of protein, which is amino acids. So if looking into this diagram, so we can see that in the mouth there is no protein digestion, because basically there is no enzyme that actually can attack the peptide linkage, and it starts in the stomach with the presence of pepsin and also renin that specifically digests protein, and continued further in the small intestine. However, the secretion is produced by pancreas, like the secretion of enzymes that focusing on protein, which are trypsin, chymotrypsin, lactase, carboxypeptidase, and also carboxypeptidase A and B. And in small intestine, it secretes aminopeptidase and dipeptidase that will further break down the peptide linkage with, to produce the end product, which is amino acid. And how protein been absorbed from the lumen into the intestinal cells. So imagine this is the lumen where the protein digestion is taking place. Protein absorption is more rapid in the duodenum and duodenum compared to ileum. About only 2 to 5% of the protein in small intestine escape digestion and absorption, meaning that majority, like about 95 to 98% of protein that been ingested will be absorbed into the small intestine and further on to the capillaries. Some of the ingested proteins enter the colon and is eventually digested by bacterial action. And protein in the stool is not of dietary origin, however, it comes from the bacterial and cellular debris. And absorption of proteins antigen, particularly bacterial and viral proteins, take place in large macrophage cells of M cells bias patches. So we can see here the transportation of sodium into the intestinal cells will produce energy, and that energy will be used to transport the dipeptides and tripeptides into the intestinal cells, and it's further digested into amino acids, and with the presence of active transportation of sodium out of the cells in the exchange of potassium that produce ATP based energy, it will be then amino acid will diffuse into the blood capillaries here. And now we're moving into the digestion of fat. One of the important steps in the digestion of fat is emulsification. This is referring to the bile salts that breaking down the large lipid droplets into smaller droplets. So basically, I need to increase its surface area for optimize the digestion. So from the one large lipid droplets, it needs to be broken down into smaller droplets, and this is performed with the presence of bile salts. It increases the surface area where lipase can act on the smaller droplets. So the enzyme that is responsible for fat digestion is lipase, and it's also secreted by pancreas. Pancreatic lipase breaking down small lipid droplets into free fatty acid and 2 monoglycerides. This is the basic compound of fatty lipid, which is fatty acid and 2 monoglycerides. And this is a water soluble called measles. Okay, free fatty acids and 2 monosaccharides enter enterocytes by simple diffusion and reassemble into triglycerides. And then these triglycerides combined with will bind to the protein and form chylomicrons. And this chylomicron is the one that's secreted across the basolateral membrane by exocytosis and enter lymphatic capillaries and also and later on to bloodstream. So different with amino acid and also glucose, fructose, and like the end product of carbohydrate just now. Fat is actually been absorbed into small intestine cells and then will be passed or absorbed into lacteal or lymphatic capillaries. Okay, so if you cellular level, we can see here, this is the fat droplets. So the bile salts will actually break the large fat droplets into small droplets, okay, and then it will further broken down by lipase to form monoglycerides and also fatty acids, and they actually bind together to form micelles. And this micelle is water soluble, so they actually diffuse into the small intestine cells, and then if we could here and combine with the protein to form the chylomicron, and then they will be absorbed into the lacteal. Okay, this is the lacteal and going to the lymph vessels and later on to the back to the blood capillaries. Okay, now we are moving to vitamin and also iron absorption. So for vitamin, for water-soluble vitamin, they will be rapidly absorbed into the small intestine cells. However, for fat-soluble vitamins, the absorption depends on the fat absorption. And most vitamins are absorbed in the upper small intestine. In exemption for B12 vitamins, it is absorbed in ileum at the end part of small intestine with the presence of intrinsic factor that's secreted by stomach. So if you still remember, in the stomach, apart from acid hydrochloric, apart from pepsin and renin, the intrinsic factors is one also the gastric secretion content, and it's actually function to have B12 absorption in the ileum. And with if there is a peptic ulcer that damage the gastric mucosal layer, it will compromise the production of the release of intrinsic factor and further will affect the B12 absorption, will result in pernicious anemia. And then for iron, free ions, which is ferrous, is absorbed in the upper small intestine in duodenum part as well. And ferrous combined to transporter, which is proprotein, and is transported into the blood. In plasma, ferrous is converted to ferrum and found to transferrin ion as a ion transport protein and bound to transferrin. Okay. So in blood, the iron present as iron with 3+ charge. Okay, with the sodium and potassium, sodium is actively absorbed throughout the large and small intestine. We can see how sodium is very important because the active transportation of sodium across against the concentration gradient is actually produce energy, and this energy is used by the end product of food to actually be transported into the intestinal cells and even to the blood capillaries. And potassium is absorbed by diffusion along blood lumen potential gradient. So for calcium absorption is actively absorbed in duodenum and require energy. So this is calcium in the lumen will be actively transported into the duodenum cells using and it's require energy. And with the active transportation, meaning that the concentration of calcium here is actually lower than the small concentration of calcium in the small intestine cells. That's why they need to go against the concentration gradients, and it needs energy. Entry across the apical brush border is mediated by the specific calcium entry channel, which is known as calcium channel. Okay. This is the calcium channel. Within the enterocytes, this is the enterocyte is the small intestine cells. Calcium binds to calcium binding protein. This is the transport protein that will transport the calcium from the enterocytes out of the enterocyte into the blood capillaries. And calcium exit across the basolateral membrane is mediated by the plasma membrane of calcium ATPase PMCA1. This is a channel that transports the protein out of the cell into the blood capillaries. And then this is regulated by active form of vitamin D. So vitamin D is important for calcium absorption. Okay. And in small intestine, there is also absorption of water. So if you still remember, the cumulative amount of water in small intestine is about 9,000 and 9 liters. This is actually coming from about 2 liters coming from the water that we ingested from drink of soup, and another 7 liters are coming from the intestine, the GIT secretion. For example, like in salivary glands, it's produced about 1.5 liters, gastric secretion 2.5 liters, bowel secretion about 500 ml, pancreatic secretion about 1.5 liters, and intestine itself secrete the intestinal secretion about 1 liters. So this altogether is about 7 liters, and out of these 9 liters of fluid of secretion, about 8,800 will be reabsorbed, okay, into the intestinal cells and back to the blood capillaries, and it left us about, and basically the distribution is the duodenum will absorb about 5.5 liters, in the ileum about 2,000, and the balance about 1.3 liters will be further absorbed in colon. Okay, so the balance about 200 ml is the one that we excrete in the stool. Okay, so these coming two slides are the summaries of the digestion and also absorption of the nutrients. Okay, so we can see here proteins, lumen of small intestine will be absorbed in the form of amino acids. Okay, and then it will be absorbed by the blood capillaries. Okay, and also the same like carbohydrates, monosaccharide referring to the glucose, fructose, okay, galactose, and then they will be absorbed by the small intestine cells, okay, and then will be further absorbed into the blood capillaries. And then for bowels, for fat, the bowels will emulsify the droplets into smaller droplets, and further broken down, the lipase will be further broken down into fatty free acids and also monoglycerides, and we absorb into the cells, combined with protein, will known as chylomicrons, and then will further absorb into the blood, into the limbs, vessels, lacteal. Okay. And this is another diagram in terms of the types of transportation. Like glucose will be secondary active transport with the coupled with sodium. Okay, and fructose facilitate diffusion. Amino acid also secondary active transport coupled with sodium transportation. Peptides also the same mechanism, same types of transportation. And for free fatty acid and measles, is simple diffusion, meaning that they actually enter the enterocytes, which is small intestine cells, following down the concentration of fatty acids. Okay, so the fatty acids concentration in the lumen is higher as compared to in the cells, so they can enter the cells via simple diffusion, as compared to amino acid and also glucose, in which the presence of glucose amino acids is lower, the concentration is lower in the lumen. That's why they need to be transported into the small intestine cells by transsecondary active transport with the presence of sodium transportation. Okay. Okay, so now we are moving into the large intestine. At the end of this lecture, I hope the students will be able to state the motility and movement of large intestine, which is colon, and then able to state the absorption process in the colon, and describe the mechanism of defecation and its control. So this is the large intestine, colon. This is, imagine this is the end of ileum or small intestine. So the first part is cecum, and then ascending colon, transverse colon, descending colon, sigmoid colon, and rectum, and also anus. So the large intestine consists of colon, cecum, appendix, and rectum. Appendix is like a finger-like projection, okay, at the beginning of colon, and it means function basically for like a limbs vessel, which is any bacteria or virus can actually be reside in this appendix. The small light finger-like projection at the bottom of the cecum is the appendix, a lymphoid tissue that houses lymphocytes. Okay. Main function for colon is for drying, meaning that for further absorption of water, and also storage, because the feces will be stored in the rectum prior defecation. The content delivered to the colon from small intestine basically is indigestible food, residues like cellulose, and also unabsorbed biliary components, and also remaining fluid. And these contents will your colon will extract more water from this content and salts, and then producing the final product, which is the feces. So we need a mechanism to actually move the food along the colon. So we have segmentation contractions and also peristaltic wave, and on top of this, the main mechanism of motility in the large intestine is mass movement. This only occurs in the colon, a simultaneous contraction of smooth muscles over large confluent area, and it moves materials from one portion of the colon to another down to the rectum. And distension initiate defecation reflex. So distension of rectum indicates that then the feces need to be defecated. Okay, and it occurs 3 to 4 times per day in normal human being. So absorption in large water mainly is in large intestine mainly is water, about 850 mils in cecum and also ascending colon. And so sodium ions will be reabsorbed in this region as well, and chloride ions. Okay. And at the same time, small amounts of glucose, amino acid, and fatty acids that not being absorbed in the small intestine will be further absorbed in large intestine. And some types of drugs like anesthetics, sedative, tranquilizers, and some steroids also been absorbed in the small intestine. Okay. Bacteria action in the large intestine. A large amount of bacteria are consumed along with food. So no matter how actually we try to make sure that the food is hygienic, clean, but we cannot, inevitable, some bacteria actually still present in our food. Some of these bacteria destroyed by acid in the stomach, but some will pass through unaffected. These microorganisms present in the colon include not only bacilli but also pleomorphic organisms and cocci of various types. Some enteric microorganisms synthesize vitamin K and a number of B complex vitamins and folic acid. And ascorbic acid, cyanocobalamin, and choline are utilized by some intestinal bacteria. So the brown color of the stools is actually pigment formed from the bowels pigment by intestinal bacteria, and bacteria also produce some gases in their flatus. So basically this is what we know as a good bacteria. They have some function, they produce vitamin K and also some B complex vitamins, and at the same time they also further they actually contribute to the nature or the features of the feces. Okay. Defecation. So the spinal reflex, defecation is regulated by a spinal reflex, which is through actually rectum and sigmoid colon are empty. Defecation referring to the empty process of rectum and also sigmoid colon. Yeah. The rectum play a role as a reservoir, like for storage of feces until the rectum is distension, the distension of rectum and or is full enough to actually giving a signal that the feces need to be defecated. Mass peristalsis, which is the same like mass movement, causes the feces to enter the rectum. Distension of the rectum initiate reflex contraction of its musculature and the desire to defecate. The urge to defecate first occur with the pressure about 18 mmHg, and external as well as the internal sphincter relax and the contents of the rectum are expelled. Okay, voluntary defecation initiated by voluntary relaxing of external sphincter and contracting of the abdominal muscles. Okay, so when the abdominal muscle contract, will further increase the pressure in the large intestine, and as the external sphincter relax, so it's allow the feces to be defecated. And then gastrocolic reflex, stimulated with the action of gastrin in colon, will increase colon motility and also stimulate urge to defecation. So this diagram, we can see that there is a distension of rectum, like rectum full feces. So afferent nerve will send a message like through the mesenteric plexus splenic nerve, we send a message to the spinal center in the lumbar sacral region. Hey, we are full here, we need to be defecated. So the afferent nerve which is S2, S3 and S4 will send a message from the spinal center to the rectum, giving a signal to contract, for contraction of the descending colon, sigmoid colon and rectum. And at the same time, there will be inhibition of internal anal sphincter. With this inhibition of internal anal sphincter, they will result in relaxation. Okay, that will allow the feces to be excreted into the anus. Okay, so what's the physical properties of feces? The amount is about 60 to 100 grams, with the average of 100 grams. It can be semi-solid or solid. Okay, in diarrhea, it's very watery because the bacteria that causes the infection that resulted in diarrhea might actually damage the ability of small intestine and large intestine to absorb water. Okay, it's slightly acidic, with the pH of 5 to 7, and the color it differ according to the situation, like yellow due to the presence of stercobilin, black in the presence of altered blood, which is known as melena. If there is a bleeding in the upper part of gastrointestinal tract, for example in the stomach, due to peptic ulcer, so by the time the blood arrive in the form of feces, mixed with food and forming the feces, it will be black in color. Pale in steatorrhea condition, like if there is a problem with the bowel secretion, then the fatty food cannot be processed, so fat will be present in the feces and resulted in pale color, and clay color in the presence of bile. Okay, odor or smell is indole and scuttle with the presence of gases and also fermentation of carbohydrates. In terms of chemical composition of feces, mainly water is about 70% of total weight, and a part of that there are cellulose, bacteria, inorganic material, fat and fat derivatives, and also desquamated mucosal cells, mucus, and small amount of digestive enzyme. Okay, so that's all for today. So inshallah, we are going to meet in the real-time teaching. I'm happy to accept any questions for further increase your understanding. So I hope that by now you already understand what is happening to your food, the one that you eat from your mouth down to pharynx and esophagus, and further down to the stomach, and now in the longest tubes of the GIT, which is small intestine and large intestine. So inshallah we'll see in the fourth lecture that covering more into details on pancreatic and also liver and biliary system. Okay, Assalamualaikum warahmatullahi wabarakatuh.